Yes. Today we are going to know about plasma cell disorder or plasma cell neoplasia. So what is plasma cell neoplasia? So it is an abnormal proliferation of immunoglobulin producing cell. So basically it is a B cell proliferation. So this result in accumulation of monoclonal immunoglobulins in serum and urine. Monoclonal means any single. So you can see any single accumulation of immunoglobulin in serum and urine. So these serum and in serum and urine you can see this abnormal immunoglobulins or elevation of this light chain or heavy chain. So what is this plasma cell disorder is also called as? So it is called as plasma cell dyscrasia, paraproteinemia, dysproteinemia and monoclonal gammopathies. So what does all this includes? So it includes 6 diseases. So what are these 6 diseases? So these 6 diseases are multiple myeloma. So multiple myeloma is one of the most commonest deadly disease which you can see in most of the cases. And next is localized plasmacytoma and lymphoplasmacytic lymphoma, Waldenstrom macroglobulinemia and heavy chain disease, primary amyloidosis and MGUS. So what is MGUS? MGUS is monoclonal gammopathy of undetermined significance. So we will going to study all about this in coming chapter. So some of the features of plasma cell disorder. As I say, this is a neoplastic proliferation of B cell lineages. So for this you have to know the normal B cell lymphoma. So normal B cell lymphoma contains both M and G heavy chain. So when these B cells are stimulated, they are exposed to the surface antigen. So what is the surface antigen? These immunoglobulin specific surface antigen. So they mature and they form an IgG producing plasma cell. So these are the normal one. So what happens in plasma cell disorder? So in plasma cell disorder, when the control is lost, there is result in abnormal production of immunoglobulin. So abnormal production of here single monoclonal immunoglobulin. It might be IgG or IgM or any other thing. So what all the changes you see in B lymphocytes? So in B lymphocytes, first there will be B lymphocytes. It starts as reactive B lymphocytes and it converts into plasmablastoma, proplasmacyte and plasmacyte or plasma cells. So how does it look different from abnormal and normal plasma cells? So in abnormal plasma cells, so it is usually bigger. Bigger means it is 14 to 20 microns and you can see round to oval in shape with eccentrically placed nucleus. So it is round, eccentrically placed with coarse chromatin, and there will be deep blue cytoplasm, and you can also see a perinuclear halo. So this perinuclear halo is because of Golgi apparatus. So what is the major thing which you see in this plasma cell? Abnormal plasma cell. So here you can see that cartwheel pattern. So what is that cartwheel pattern? Is like this, which is a clock face, clock like structure. So you can most commonly appreciate in formalin fixed tissue, but sometimes also you can appreciate it in normal peripheral smear, which is more evident. So the basically the thing of this is the immunity is lost when you can see. And next is so as I said, there is monoclonal proliferation of IgG and there is excess of light chain. So light chain may be kappa or lambda. So these are the free light chains. So these free light chain get elevated. So when this is elevated, they show marked towards either kappa or lambda. So any one you can see the elevation. So because these free light chains are very small and very small in size, they get excreted in urine. So these are referred as Bence-Jones protein. So these are the free light chains which can be determined in urine. And you can also see the elevation in heavy light chain, either alpha, gamma or IgM. Okay.